Prior to performing any forms of cell culture, ensure that the BSC is UV'd for at least 10 to 15 minutes. At the same time, any equipment that is required to be in the BSC has to be sprayed and wiped down with 70% ethanol. And you have to put on the appropriate PPE, which includes a lab coat as well as a pair of latex gloves. Before performing cell culture, you must ensure that you wash your hands thoroughly. And as mentioned earlier, you require the appropriate PPE, a lab coat, as well as a pair of latex gloves. Also remember to spray and wipe down the hood with 70% ethanol to sterilize the working area. Make sure to spray and wipe down all equipment that is to be used inside the biosafety cabinet. It is important to always warm up your media before adding them to the cells. So this is what your hood should look like. And not like this. Moving on to cell culture. Do not pour away the media directly into the bin. Instead, use a sterile serological pipette along with the pipette boy to remove the media into a waste container. After emptying the media, PBS is added to wash the cells. During the washing process, do not shake the flask vigorously as shown. Instead, tilt the flask back and forth gently to perform the washing. In cell culture, aseptic technique must be observed at all times. As such, you must not use the same pipette tip for the different solutions you use. After incubation with trypsin, do not do this. You should tap the flask gently to detach the remaining cells that adhered to the flask. Ensure that the serological pipettes do not touch anywhere except the solutions you use, such as media, trypsin, or PBS. You should neither paraffin nor tighten the cap of the cell culture flask before placing it into the incubator. Loosen the cap slightly so that carbon dioxide can diffuse into the flask easily.